Hello, everybody. I'm Paulette Evans, and I'm super excited to be leading this AP Biology study session with you today. And the topic for this session is going to be bioenergetics. Now, before I get started, those that know me know I always like to start with just a little bit of a temperature check on you to see how you're feeling and how you're doing. So just take a couple minutes, either think to yourself or just jot down a couple of notes. How are you feeling today? How have you been doing this week? Nice, nice, nice. I hope that you're doing well. That is my hope. I think, often when I ask students, what topic in AP biology they feel stressed or really tight around, bioenergetics generally rises to the top. And I think just because the main two processes, photosynthesis and cellular respiration, can seem kind of overwhelming, it is my hope that this session kind of helps you get connected to some kind of foundational conceptual understandings of this process. And what I mean by that is kind of brass tacks, like what's happening in the processes in a kind of a general sense, in a way that you're successful on the AP biology exam without kind of overwhelming you with minute details that are important. They are important, but that you don't need to like subject to memory at this point. Okay. So, again, welcome to this AP biology session focused on bioenergetics. I bet you've seen a picture similar to this before. What would you call this? You're probably thinking like a food chain or a food web. You're absolutely right. Absolutely right. And so there's two main points that I kind of want to take away from this picture, although there are lots of things to talk about here. The first thing I want to talk about is the flow of energy, the flow of energy. And so you see in this particular ecosystem, the energy is going to start with the sun. And you can hopefully you can see my cursor here starts with the sun. And then there's a collection of organisms that can bind that energy from the sun and then transform that into organic molecules. And that group of organisms are called autotrophs or producers, as you see here. Okay. If you notice here, every time that there is an energy transformation, once you take energy from energy coming in as photons of light to energies in an organic molecule, let's say glucose, you're going to have energy lost into the atmosphere, into our universe as heat. So every time you change energy's form, you're going to lose a metabolic process, or in this particular process, which we would call photosynthesis, you're going to lose heat to the atmosphere. All right. And so here you see this next kind of collection of living organisms that are called consumers. And if you remember from some of your classes, you've got primary, secondary, tertiary, quaternary consumers. So primary consumers will be able to consume, will probably be consuming producers directly. Then you have other levels of consumers that can do various types of eating patterns. They can eat other consumers. Sometimes they'll eat producers as well, but that'll be more energy transformation in those organisms. Just know there'll be more energy lost as heat in those stages as well. But what's important to recognize here is that energy flows through the ecosystem, all right, where a majority of the energy is lost as heat. Every time we do these fancy chemical reactions, we're going to lose a lot of energy to the universe or to the atmosphere as heat. Okay, so that's the first thing. The second thing that you may see in this picture that's also marked with these arrows is nutrient cycling. And what does that mean? I'm going to try to make that very, very simple. It means all the carbons, hydrogens, oxygens, nitrogens, phosphorus that we're using in these organic and inorganic molecules, they're going to be recycled in the ecosystem. So we'll break them apart, we'll put them back together, build them back up into large molecules, we'll break them apart again. So nutrients themselves, or the actual, I think sometimes I think about the things that are found on the periodic table, the elements and the molecules that we deal with in the periodic table in this particular chapter or unit will be recycled in the ecosystem. So energy flows, nutrients cycled. All right, so this page here just kind of captures a little bit of what I said. So bioenergetics is the field of biology that concerns itself with energy flow through living systems, right? It's super important. How are living systems capturing energy? And then how are they actually using that energy? Also important, all right? All living systems require a constant input of energy. Constant input of energy. I always ask students, why is it that you get up in the morning and you have to eat? And then a couple hours later, you're kind of hungry. And then you need lunch. And then what time's dinner? What am I eating for dinner? If you think about it, we consume lots of energy by way of our food. It takes a lot of energy to keep us going as living systems. All right. And I am being a little bit kind of human focused here, but it's true for a lot of other organisms as well. All right. Living things use chemical energy stored in the molecular bonds of ATP, and ATP is a super important molecule that we'll talk about in just a second in more detail, and other organic molecules to perform life's functions. All right. Sunlight is the main or original source of energy. Main or original source of energy. Autotrophs, like we just saw in the picture before, or producers like plants, algae, and some bacteria, capture energy from the sun or from inorganic chemicals in some cases, and transform it into sources of energy that are usable by cells. Okay, so here, this first energy transformation by autotrophs, right, is photosynthesis. Is photosynthesis, and then the secondary energy transformation is like taking the product of photosynthesis, which is glucose, and then transforming that energy source in glucose into energy source in this molecule that we just talked about here, ATP or adenosine triphosphate. These sources are often either ATP or sugar molecules, which I just said. So sugar molecules, glucose, you're going to hear a lot in class, or ATP, although we can metabolize lots of different molecules besides sugar. We can metabolize sugars, like glucose is an example. We can metabolize proteins, and we can also metabolize fats as well. And sometimes students ask about DNA. I guess you could metabolize DNA, but we shouldn't be doing that. So no. So proteins, sugars. We'll talk a lot about sugars and fats or lipids. All right. Also, I talked about in that first picture, this is just a summary, if you will. Matter cycles continuously through an ecosystem. The matter or the atoms and the molecules, the pieces, always remember the things on the periodic table, the carbons, hydrogen, oxygen, nitrogens. All right. So they're used over and over in the ecosystem. All right. And energy flows through the ecosystem. Okay. There's a constant input of energy into the ecosystem from the sun, always and constant. This energy is transferred and transformed at each level of the food chain or the food web and is eventually released as heat or waste energy. And we're going to talk about the kind of the importance of that in just a second. All right. The heat does leave the ecosystem. And this is also a mechanism for keeping endotherms warm, this type of pathway where we kind of with each kind of metabolic reaction, we have heat being produced that also keeps us warm at a constant temp inside of our body. We still do lose a lot. We still do lose a lot of heat. All right. An extremely important idea from this unit is that the highly complex organization of living systems requires a constant input of energy and the exchange of macromolecules and I named the big three classes that will be kind of important and central to these conversations around photosynthesis and cellular respiration, sugars, proteins, and lipids. Thermodynamics. Okay. Those very connected to what we were just talking about with the food chain, food web, and then the content slides we just looked at. The first is the first law of thermodynamics. And here it's clearly stated energy can be transferred or transformed, but neither created nor destroyed. Cannot get rid of energy, right? You can only change its form. And so I'll use the first example. So from the sun to autotrophs or producers. Sunlight travels in photons. So that's sunlight energy or solar energy, right? That's its form. And then photosynthesis is the process that autotrophs or producers are going to utilize to capture the sun's energy, take that energy and transform it into the energy into sugars. All right. Organic molecules, this particular organic molecule or macromolecule, a sugar, or you may say glucose. All right. And so that's the first law of thermodynamics. Energy can be transferred or transformed, but neither created nor destroyed. Very important. Now, I love this picture and I love this whole picture, but I love this picture with the big cat because I think it, in a beautiful way kind of describes the second law of thermodynamics, which can be a little bit tricky. The second law of thermodynamics is every energy transfer or transformation increases the disorder or entropy of the universe. All right. Entropy or delta S, if you're familiar with Gibbs free energy equation here. So here you see that there, let's go back to this first picture really quick. You see that you have the big cat eating some prey. And so the proteins inside of the muscles and the fat inside of the organism are being consumed by the big cat. The big cat is going to break those those proteins down, and it's going to extract that energy and transform that into the energy of ATP. All right. And so now that you've eaten your food, you've changed it over to ATP, now you're going to do life's work. Right. And so you see now here that same big cat is on a hunt. So that big cat is now using the energy from this kill to go and hunt some more. And so what's happening is, as it's breaking down those organic molecules from this deceased organism, it's going to break down those sugars into CO2 and the H2O in the process of cellular respiration to get ATP, right? That energy is going to go from the proteins and the sugars and the fats inside of that deceased organism, and we're going to change that over or transform it into the energy of ATP. That process is going to require us to break down those organic molecules into CO2 and water, and you're going to lose a lot of that as heat. So what is the organism getting here? ATP. But as you break down the organic molecules from the deceased organism, you're going to break them down to their component parts, CO2 and water, to get the ATP, which the organism is now using, and you're going to lose a lot of that energy as heat. So if you take large molecules, break them down to get the energy out of them, you're going to breathe some of that out, CO2 and H2O, you're going to breathe that out. Taking large molecules, chopping them up, breathing them out, notice how that creates more disorder outside of the organism. The organism itself is still more ordered. The organism is doing its job, is going on a new hunt. It has made some ATP through that energy transformation, but it's broken down a lot of molecules in the process. Those broken bits of the molecule are gone back out into the atmosphere. It's making the atmosphere around it, or the universe around the organism, more disordered, more bits and pieces of molecules around it, if that makes sense. I hope that makes a little sense. Also, this release of heat is also creating more of a disordered space around the organism or the universe. When we say the universe, we just mean the atmosphere outside of the organism. Okay. So with each energy transformation, so in particular here, cellular respiration, when this organism is breaking down organic molecules, I don't care if it's a sugar, a protein, or a lipid, but as this organism is breaking down organic molecules for that energy transformation into ATP, you're going to break apart these larger molecules into smaller ones, releasing them to the atmosphere along with that release of heat. And it's going to create more of a disordered atmosphere around the organism. All right. So increase of delta S, or an increase of entropy. Entropy. There's the word you want to remember. With each energy transformation, you're going to increase delta S, or increase the entropy of the universe around you. All right, and here's just a summary. All right, thermodynamics is the study of heat, energy, and its properties. And be sure to capture these slides if you need to. The first law of thermodynamics, also called the law of conservation of energy, is that energy cannot be created nor destroyed, but it can be transformed from one type of energy to another type of energy. All right, and can be transferred, from one location to another location. Energy is never used up. And we talked about that a couple of slides ago, because we'll have a constant input of energy. As long as the sun is beaming on us, and as long as, for us humans, all right, as long as we have autotrophs or producers that can bind that beautiful energy from the sun, all right, we will never use energy up. All right. The second law of thermodynamics says that during each energy transformation, some of the energy is lost as heat, and you're also going to be losing smaller pieces of broken down molecules. These transformations increase the disorder or entropy of the universe around the organism. All right, metabolic pathways. All right, and this kind of goes into what I was just talking about with the big cat. Okay, all right. So two different things. We have two basic types of metabolic reactions. You can have an anabolic reaction or a catabolic reaction. Let's look at anabolic reactions. Anabolic reactions start with smaller molecules, smaller component parts, with a net infusion of energy. So with energy and smaller molecules, you can push those molecules together into larger molecules. So energy in to build. If you want to build a large molecule, and I can give you an example. So if you want to take monosaccharides and you want to stick monosaccharides together to build a polysaccharide, then you don't have to have a net input of energy to build anything. If I wanted to build a building, you've got to put energy into it. It's not going to build itself. Am I right? You got it. Same thing with amino acids. If you have small amino acid molecules and you want to build a large protein, you're going to have a net infusion of energy to build that. All right. So energy in small molecules to build larger molecules. Okay. Now, the converse of that is a catabolic reaction. A catabolic reaction is breaking apart large molecules. All right. So if you have a huge polypeptide or a huge protein molecule and you break that molecule apart, you're going to have a release of energy, and then you're going to break those molecules down to their component parts. And that's synergistic with what we were talking about, especially this catabolic reaction with the picture just before with the big cat. All right. And here's the content slide that goes along with that. Metabolism is the sum total of all of the chemical reactions going on inside of a living organism. All right. All of the life's processes occur due to chemical reactions. All of these are chemical reactions, most often multi-step reactions. First, here you have catabolism. This refers to chemical reactions which break down molecules. And remember, there is a release of energy in these chemical equation or exergonic reactions. And then you have anabolic pathways, and anabolic or anabolism or anabolic pathways. These reactions combine monomers to build polymers. And so I was specific in the last slide. Monomers are monosaccharides, small pieces. You need energy to put them together to build large sugars. All right. Amino acids are monomers that if you want to put them together to build a protein, you have to have net input of energy. All right. It also it doesn't work quite the same for lipids because they don't build long chains. But anabolic pathways require a net input of energy, and those are the two big groups that we'll talk about: proteins and carbohydrates for this particular session. All right, metabolic pathways and biological systems are sequential. All right, the product of one reaction serves as the reactant for the subsequent reaction. The sequential nature of these pathways allow for controlled and efficient transfer of energy. Many of the metabolic pathways in living things are shared across many, if not all species, due to common ancestry. We're going to talk about common ancestry quite a bit, and the AP exam relies quite a bit on common ancestry. We'll see lots of multiple choice and maybe even a few FRQs that rely on this sense of, if most organisms can do a thing, what does that actually mean? All right, and so here's an example of a metabolic pathway. And so here you have the start molecule A. So start molecule A is going to be converted into product B. But how do we get to product B? It is our first reaction, but enzyme 1 is going to serve as the house, if you will, for this first reaction or reaction 1. So literally how that happens is most often is start molecule A fits into the active site of enzyme 1. Enzyme 1 is the house of the reaction, so it helps lower the activation energy and just make all of the conditions, environmental conditions, conducive for the reaction to occur. And then what comes out of the active site of enzyme 1 would be product B. Notice here that product B now is serving as the reactant for reaction 2. And so now which was product B is now reactant B. B is going to fit into the active site of enzyme 2. And then what's going to come out of enzyme 2? I'm going to let you guess. C. You got it. C comes out of enzyme 2. This is most often how this will occur, okay? And why enzymes are important, primarily because they lower the energy of activation for the reaction or reaction 2 in this case, and they also make several other environmental conditions just perfect for the reaction to occur. And the last one in this particular example, you have product C, which is now reactant C, which will fit nicely into the active site of enzyme 3. Enzyme 3 will do its thing, and then what comes out of active site for enzyme 3 would be product D. All right. You'll sometimes see questions like this that will also talk about negative feedback or inhibition of enzymes. And I'll give you an example just really quickly of how that would work. All right. Sometimes product D is your final product here. Sometimes product D will regulate the quantity of itself. So let's say you're going through reaction one, two, and three, and you're making a ton of product D, a ton of it. Do you need to continue to make D? Maybe not. So, what do you do, or what could you do to stop the production of D? In a lot of cases, not all, but in a lot of cases, D can stop the production of itself. And it would do that by acting as a competitive inhibitor or another type of inhibitor. It could be an allosteric site, but it could be act as an inhibitor at one of the enzymes. So, we'll use enzyme one. If there's too much D, D can sometimes compete with A for this enzyme 1 active site. So let's say that D gets into the active site of enzyme 1 instead of A. What essentially would happen is you would shut the pathway down because only A can get you B. D inside of enzyme 1 will not get you B. Okay, but D can fit. But just because D can fit doesn't mean you're going to get product B. The only way you get product B is if A goes into the active site of enzyme 1. But if D, in my example, if D can compete for the active site for enzyme 1, then D can then inhibit or stop the production of itself, which is a pretty cool feature. All right. And that's called negative feedback or inhibition of a metabolic pathway. All right. All right, so metabolic pathways are enzyme regulated sets of biochemical reactions that lead to either biosynthesis or anabolic pathways that require energy, or breakdown of catabolic pathways that will have a net release of energy. Examples of metabolic pathways include glycolysis, Krebs, and the Calvin cycle. The first two, glycolysis and Krebs, are in cellular respiration. The Calvin cycle is inside of the light independent reactions or inside photosynthesis. Photosynthesis and cellular respiration and digestion might also be thought of as metabolic pathways. Each step is catalyzed by a different and specific enzyme. Specific enzyme. All right. So I know in that previous example it was like enzyme one, two, three. It's because it was a generic example. Just know that these enzymes have actual names and they do a very particular thing. All right. Since enzymes are proteins, these enzymes are coded for by the DNA. Shared and conserved sequences of DNA lead to similar enzymes and similar metabolic pathways across living organisms. This is evidence of common ancestry. You see that common ancestry again. A lot of times in these sessions, I like to shout out one protein sequence or DNA sequence that the AP writers talk about a lot or include a lot in their exam, and it's cytochrome C. So either talk about cytochrome C DNA sequence or the cytochrome C protein that's produced from the DNA sequence, but they use that a lot in a lot of multiple choice and sometimes in the FRQ. And the reason why they use cytochrome C a lot is because cytochrome C is highly conserved. That DNA sequence is highly conserved, which means that very simple organisms have it, very complex organisms have it. It's been around a very long time, millions of years. So if you want to compare organisms, processes between organisms, talk about relatedness between organisms, you're always going to pull for molecules or proteins and DNA sequences that a lot of organisms share. Otherwise, you couldn't compare them. And so cytochrome C is just a great example of common ancestry, not in a process, but in a particular molecule that's encoded in our DNA. All right. So moving forward just a little bit, I want you to think to yourself, which molecule is the primary energy currency in the cells of living organisms? Okay. What is the molecule that we are going to pull for to do cell work? For me to yap right now, for us to breathe, for us to think, for us to move? Which is the molecule that we're looking for in living systems? You guessed it. ATP, or adenosine triphosphate. Let's look at the basic structure of adenosine triphosphate because I'm telling you right now, we recycle our parts, we recycle our elements. Okay. Remember, I told you the carbons, hydrogens, nitrogens, we recycle those bits. Okay. So ATP is going to look like something that you've probably seen before. All right. So let's look at the parts here. First, you have this molecule here. This is one molecule of ATP, but I'm just going through the component parts that can exist as kind of either a functional group or molecules on their own. But this is one complete molecule is bound together. But one component piece here is adenine. Adenine. And some hallmarks of adenine is you see nitrogen littered through there. You don't see carbons because carbons are at these vertices that don't have the letter N on them. Those are carbons. Okay. So where have you heard adenine before? Adenine. Ooh. A to T, C to G. Am I right? From DNA, right? A to U, C to G, RNA, but that A is consistent. The A in DNA and RNA is adenine. And so here you see that adenosine triphosphate, part of it kind of looks like a little bit of DNA. And that theme continues. Let's look at the second part here. The second part of the whole ATP molecule, remember it's one molecule, is a sugar. Which sugar is it? Do you see in the picture? Ribose sugar, ribose sugar. Now, which nucleic acid, this is ATP, but which nucleic acid has ribose sugar? DNA or RNA? You got it. RNA. So just from these two pieces here, you've got the sugar that's in RNA. You've got one of the nitrogen bases that's in RNA. Let's take a look at the last piece of ATP. Three phosphate groups. Well, listen, if you take off two of these phosphate groups, that is a nucleotide from RNA because it's ribose sugar. So that is one nucleotide from RNA. So ATP is essentially a nucleotide from RNA plus two phosphate groups. Plus two phosphate groups. Okay? This is the molecule that you are using a lot of right now to power your entire body. Everything that you're doing inside of your body, from the chemical reactions that you don't feel to the things that you can feel and can think about, being powered by adenosine triphosphate. Okay. The two big energy bonds here, you see they're kind of marked with these squiggles here. These are high energy bonds here. So as you cleave these phosphate groups, you extract energy from these bonds. And we'll talk a little bit more about that in just a second. The reason why these are high energy bonds, and if you've had chemistry, this will make sense to you. Phosphate groups are negatively charged. You can see the oxygens here on the outside. They are negatively charged in these phosphate functional groups, the phosphate groups. Negative groups do not like to be next to negative groups. Y'all know how charges attract. Opposite charges attract. Plus attracts minus. But if you have negatively charged functional groups that are relatively large, they do not like sitting together. So it takes a lot of energy to push them together in this molecule. And it's going to release, a lot of energy when you break off this 2,3 bond and the 1,2 bond between the phosphates. All right, so that's adenosine triphosphate or ATP. It's a modified RNA nucleotide. And so it's an RNA nucleotide with two extra phosphate groups. And then you have a high energy bond here on these two last phosphate groups. You break them apart or break them off, you can extract a lot of energy from ATP to do cell work. All right, there's that picture again. We've talked about that structure. All right, so here's the content slide. I'm going to read through it with you. Adenosine triphosphate or ATP is a small, relatively simple molecule. It can be thought of as the main energy currency of cells, much as money is the main economic currency in human societies, most human societies. ATP is not a long-term storage molecule for chemical energy. That is the job of carbohydrates such as glycogen, which we'll store in our liver, and fats. All right. When energy is needed by the cell, it is transferred from storage molecules into ATP. All right. That process is going to be cellular respiration. And remember, I told you that carbohydrates can go into cellular respiration, fats can go into cellular respiration, and proteins can go into cellular respiration. ATP then serves as a shuttle delivering energy to places within the cells where energy consuming activities are taking place. The energy released by the hydrolysis or breakdown of ATP, the conversion of ATP to ADP or ADP to AMP, the monophosphate, is used to power most of the body's energy requiring cellular reactions. ATP is able to power cellular processes by transferring a phosphate group to another molecule, a process called phosphorylation. Phosphorylation. This transfer is carried out by special enzymes that couple the release of energy from ATP to cellular activities that require energy. All right. So let's take a look at energy coupling. All right. So here's the ATP cycle, and this is energy coupling, or excuse me, ATP coupling. Call it energy coupling or ATP coupling. All right. So let's start with adenosine triphosphate. All right. So there's your adenine, your ribose, your three phosphate groups. That is ATP. All right. So it says energy for endergonic reactions. All right. So reactions that need energy, a net input of energy. Well, where are we going to get energy from? We're going to get energy from stripping off one of these phosphate groups, right? Phosphate itself can then affix itself to another molecule, destabilizing that molecule, thus kind of starting a new chemical process, right? Or activating a new chemical process. That phosphate, remember, is large and is negatively charged. If you take it off of ATP and sticking it on some other unassuming molecule, you can change the three-dimensional structure of that molecule and activate a whole nother pathway. Okay. All right. So basically, you've used ATP to start another reaction that required a net input of energy, a net input of energy. All right. So then the question is, how do we then restore our adenosine triphosphate? Because now, because you've broken off a phosphate group, you now have adenine, ribose, and two phosphate groups, or adenosine diphosphate. Adenosine diphosphate. Well, we just need to add a phosphate group back to get it back to ATP, right? So in order to do that, you're going to have to get a net input of energy to construct ATP. And this is going to be cellular respiration, where you're going to be making more ATP in cellular respiration. And so there's going to be a net input of energy from organic molecules, and you're going to use that energy, and some electrons as well. We're going to talk about electrons and redox reactions to then connect this third phosphate group back to ADP and reestablishing adenosine triphosphate. Okay, so getting ADP back to ATP, you'll see a lot of that. It does happen in photosynthesis, by the way, but you'll get a large amount of ATP produced in cellular respiration. And it's important for me to say it now and for you to remember that cellular respiration is a process that all organisms will do. So I'm speaking about cellular respiration, all living organisms. So I'm saying that pretty generally, not photosynthesis, but cellular respiration. I'm speaking about it very generally, because most all organisms will do cellular respiration, some components of it. All right. And so here is the reaction coupling. Okay. The energy from ATP is often used to power endergonic reactions. Let's make sure that I'm not in your way, via a process known as reaction coupling. During coupling and energetically favorable or spontaneous exergonic reaction like ATP hydrolysis. Remember, the reason why it's spontaneous and favorable is because you've got three negatively charged phosphate groups that already don't want to be by each other. So it's not going to be too hard to strip one off. It's actually pretty easy. And that's why it's our energy currency. It's so easy for us to use it. Okay. All right, is directly linked with an energetically unfavorable reaction or endergonic reaction. So a reaction that's needing to occur but requires a net input of energy, we're going to use the energy that we extract from ATP and that kind of destabilizing nature of taking a phosphate group and sticking it on another molecule. We're going to use that to power endergonic reactions. This essentially means that a reaction that releases energy, like the hydrolysis of ATP, is often coupled with reactions that require an input of energy. All right. And you see a picture here that describes that process. Photosynthesis. Yeah. Yeah. Photosynthesis functions to convert solar energy into chemical energy. It's a super important process for us. You want to know why? We cannot do it. So for us, it's extremely important that this process occurs and continues to occur. All right. And to store the energy in the bonds of glucose molecules or sugar molecules. Okay. So twofold. All right. Capturing energy and transforming that energy from the sun into the bonds of organic molecules and chemical energy, and to store that into the bonds of sugar, right? It's a carbon-based molecule. Eukaryotic photosynthesizers like plants and algae are thought to have evolved from photosynthetic bacteria or prokaryotes like cyanobacteria. So, another extremely important point, knowing what types of organisms can do photosynthesis and which ones cannot, right? All right, so autotrophic organisms or producers, autotrophic organisms that can bind energy from the sun and transform that into organic molecules, eukaryotes like plants, and protists like algae, photosynthetic algae, and plants. That's two groups. Then you have photosynthetic bacteria or prokaryotes, bacteria prokaryotes like cyanobacteria. Cyanobacteria. So there are bacteria that can do photosynthesis. Algae or protists, some can do photosynthesis. And you all know that plants can do photosynthesis. So be very clear on which types of organisms can do photosynthesis and which ones cannot. Can you? No. Location, location, location. Location is super important when we talk about photosynthesis and cellular respiration. Here, photosynthesis. So here you see that this is the organelle, the chloroplast, and the chloroplast would be inside of the cell, inside of eukaryotic photosynthesizers, not in cyanobacteria. You will not find a chloroplast in cyanobacteria. Why? Cyanobacteria are prokaryotes and they do not have membrane bound organelles, right? So they don't have this, all right? But they do have the pigments, they do have all the membranes that are associated. So we'll talk about photosynthetic prokaryotes and how they get this work done. But when you look at a picture like this and you're looking at a chloroplast, so if you see the word chloroplast, know that you're talking about either a eukaryotic algae or eukaryotic plants. Okay, not bacteria, not prokaryotes. Okay, so here you can see that there's an outer and inner membrane, and then on the inside you can see some of the features. None of these that you need to remember. I just want to get you to the right place. You got a larger cell, right? And you know you've got mitochondria, you got the nucleus. You better have the nucleus, right? You got the endoplasmic reticulum. You got all kind of cool things happening in this very complex eukaryotic cell. Also in photosynthetic eukaryotic cells, you're going to have this organelle as well inside of the cell, the chloroplast. Okay, and so here you're going to have the thylakoids, and these thylakoids are membrane wrapped, so it's like lots of fat everywhere. Because y'all know we're mostly water, so having these kind of lipids in our membranes help to create the barrier that creates the organelles or the cell membranes. And so here you have these thylakoids that are then stacked. This is the site of photosynthesis in eukaryotes. All right, photosynthetic eukaryotes, photosynthesis, excuse me, is happening across this thylakoid membrane. I just want to point you to the place. That's all. I want to point you to the place. All right. And so here, if you look at the shape here, this is photosynthesis or part of photosynthesis, the light reactions, light dependent reactions. If you look at the shape here of this double layered membrane here, I'm going to just go back. That is the thylakoid. That is mimicking the shape of the thylakoid. Just to let you know where we are. Just to let you know where we are. Okay. This looks crazy, doesn't it? I know. It looks a little complex. I'm going to try to simplify it for you. This is not hard, okay? I'm going to simplify it for you. This is the light dependent reactions. This is going to be the set of reactions where we're going to trap or capture solar energy from the sun. And it's going to be the first step to converting that into chemical energy found in the bonds of organic molecules. Okay. And I'm going to tell you right now, you just need to be able to track the electrons. The electrons are a big part of this story, kind of the main part of this story. Okay. The electrons. Okay. So just follow me. I'm going to walk through it. And hopefully you can follow up with your course instructor with questions that you may have about this. Okay. So here you have proteins that are embedded in the thylakoid membrane. Okay. And then you see these little green pockets inside. I'm gonna simplify just a little bit because it can get a little needly. Okay. But just know that that symbolizes that there are pigments in there or chlorophyll, which gives plants and some algae their green color. All right. So that's what they're mimicking. There is a pigment called chlorophyll. Okay, that gives plants their green coloring. Now, inside of chlorophyll, there is inside the molecule itself of chlorophyll is magnesium. All right, the element magnesium. And magnesium has two valence electrons or two electrons that sit on the outer shell. Pause. Y'all remember electrons, if not from high school chemistry, from eighth grade physical science, right? There are shells, right, that have electrons in them. And then you have the last shell on particular elements, all right? And on magnesium inside of chlorophyll, there are two valence electrons, okay? Light hits chlorophyll. I'm doing a little bit of simplification, but you're going to get the point. Light hits chlorophyll. And then when light hits chlorophyll, the two valence electrons in magnesium get excited away from the chlorophyll molecule. Gets excited away. So two electrons, I'm not going to count electrons with you, but in this case, since we're starting two, those electrons will be pushed up, all right, to a primary electron acceptor. Don't worry about that. But this gold arrow here is tracing those electrons moving through from photosystem 2 to photosystem 1. All right. So the first step, light hits chlorophyll and two electrons in the valence shell or the valence ring in magnesium are excited away. That means magnesium inside of chlorophyll has been oxidized, it's lost its electrons. All right. And then the primary electron acceptor, whoever received the electrons, is reduced. So this is just pushing electrons. That's all we're doing. And why did magnesium give up its electrons? Because the energy from the sun excited them away. Okay. And so here you see that the electrons will move to this series of proteins, plastoquinone, cytochrome complex, plastocyanide to photosystem 1. And then you'll see light is going to excite magnesium again. It's going to give up the valence electrons again, and then to another primary electron acceptor. And you'll see, just follow the arrow. Those electrons are going to go to ferredoxin, then to NADP plus reductase. And once the electrons get to NADP plus reductase, when you attach electrons here, you'll have a negative charge. Then you add a hydrogen. Then you have NADPH. Y'all, all we've done is moved electrons. Where did the electrons come from? They came from that green pigment you see when you go in the grocery store and you see spinach. That chlorophyll gave away its electrons. Those electrons got pushed through these proteins in the membranes. And I can talk to you about how the electrons move or how scientists believe the electrons are moving. But we've pushed these electrons down and we've given the electrons to an electron carrier, NADPH. So the electrons started with magnesium, they ended with NADPH. And so if I were to do the redox, you would say magnesium was oxidized, the electrons went to the primary electron acceptor, the primary electron acceptor was reduced. And then when the electrons passed from the primary electron acceptor to plastoquinone, the primary electron acceptor was oxidized, it lost its electrons, and plastoquinone was reduced because it gained electrons. And so if you go to this final step, you'll say that NADP plus reductase lost the two electrons, and then the NADPH, it catalyzed that reaction for NADPH to hold the electrons to be the final kind of reduced molecule at the end of the light dependent reactions of photosynthesis. That was a lot. That's the first thing that I want you to track, electrons. Light from the sun excited electrons from magnesium, and we've captured those electrons. And if you know from chemistry, now that we have electrons, we can build molecules. If we got electrons, we can build molecules. All right, now let's talk about some other things that are going on. Because remember, there's a constant supply of energy. We got to have a constant source of electrons because there's more light. Light is coming in, light is coming in, light is coming in. But we gave up our valence electrons in magnesium. How do we get them back? How do we get them back? You get them from water. You get them from breaking apart water. I always ask students, why do plants need water? That's a great question. Why do plants need water? And you might say, plants need water so they can grow. That's true, right? But if you want to be the smartest kid in the room, you say that plants need water because water replaces the electrons to magnesium inside of chlorophyll. That's why plants need water. Okay. So what happens is Water is broken apart. You're going to look, you can see in the picture, going to extract two electrons, and those electrons are going to be replaced to magnesium and chlorophyll. And what's going to happen? Light is going to hit magnesium and excite the electrons, and they're going to go all the way through this process again. Okay. But when you break apart water, what happens when you break apart water? When you break apart water, you're going to get hydrogen ions. And so here you've got H plus ions building up in this thylakoid space. Okay. That's the first thing. So by splitting water, you're going to get a buildup of hydrogen in this thylakoid space. Also, you're going to get some oxygen, O2. Y'all, oxygen is just a byproduct of photosynthesis, it's almost a waste product of photosynthesis. I know we need oxygen. We need oxygen real bad. We need it for cellular respiration. So do plants, by the way, need it for cellular respiration. They don't need it for photosynthesis. Okay. So it is kind of just a byproduct of this process. And so O2 gas is going to escape these membranes and just be released. Okay. So you're going to do hydrogen ions, which will build up in this thylakoid space, and then O2 that'll just be released into the atmosphere. Okay, that's the first thing. Also, as electrons are moving, let's talk about how the electrons are moving. The electron scientists believe these electrons are moving in these redox reaction because these subsequent proteins, so as you go from plastoquinone to cytochrome complex to plastocyanine to photosystem 1 to ferredoxin, that each of these progressive proteins are slightly more electronegative than the one before it, which means that they have a slightly stronger pull for electrons. This one pulls for the electrons more than this complex, more than plastocyanine, more than cytochrome complex. So it pulls the electrons this way. While you're doing that, because electrons have a negative charge, scientists believe that you also have positively charged hydrogen ions that exist in the stroma outside of the thylakoid that will be pulled down across the membrane into the thylakoid space. The net effect here is that you get a high concentration of hydrogen ions in the center of the thylakoid space and a low concentration of hydrogen on the outside. And using a chemistry definition, high hydrogen is an acidic environment. So you have high hydrogen or an acidic environment inside of the thylakoid space, a more basic environment or low concentration of hydrogen on the outside. Okay. This is very important because we're going to make some ATP. It's going to be your first introduction to making ATP. Here's the protein ATP synthase. Okay. What happens? ATP synthase is going to be kind of the house for this reaction. The positively charged hydrogens that have built up a high concentration on the inside of that thylakoid space are going to barrel through ATP synthase, all right? That's called proton motive force. And we believe that that actually is a physical motion in that ATP synthase protein molecule. And that energy that's created is going to be the energy required to affix or connect a phosphate functional group to ADP. All right, so forcing a phosphate group to ADP, rendering it ATP. Okay. So how did we make ATP? We can work it backwards. Where did the energy come from to affix the phosphate group to ADP, making it ATP? Well, it was the protons barreling through ATP synthase. Well, why did the hydrogen ions barrel through ATP synthase? Because they wanted to go back to balance, right? We built up a high concentration of hydrogen ions in the center of that thylakoid space and a low concentration on the outside. And we know cells don't like that. We like homeostasis. We like balance. And so to get back to balance, they barrel through that ATP synthase. And this is a motor protein. This protein will have physical motion, and that energy created from that will phosphorylate ADP to ATP. It's pretty cool, huh? So the two molecules that will go to the light-independent reactions of photosynthesis are NADPH, carrying our electrons, and ATP, which is carrying our high energy phosphate groups, or bonds between our phosphate groups and ATP, which are going to be used in the Calvin cycle or the light independent reactions to build our sugar. All right, so let's take a quick look at the Calvin cycle or the light independent reactions. Okay, so notice here that the gas CO2 is going to be coming into the light independent reactions or the Calvin cycle, and it's going to be bound, or it's going to be fixed to this pre-existing molecule, ribulose bisphosphate. And the enzyme that's going to do this for us or be the house for this reaction is Rubisco, okay? And so I'm not going to get in the weeds here, okay? Just know that that's how we're going to take CO2 from the atmosphere and affix it to a pre-existing molecule. Notice here we're going to use our ATP to kind of destabilize the molecule. You see that other phosphate group being added here. You're going to see a donation of electrons so you can have bonding power to build your sugar around, and you'll get your first small sugar product, which is glyceraldehyde 3-phosphate. It's only a 3-carbon sugar, so you do need 2 turns of the light independent reactions or the Calvin cycle to construct a molecule of glucose. So the second phase here is reduction or donation of electrons to the sugar for bonding power. Then you have phase 3, which is regeneration of that original carbon dioxide acceptor, ribulose bisphosphate. So it's very simple. Bring in CO2, attach CO2 to a pre-existing sugar-like molecule. You're going to use ATP to kind of destabilize and activate the pathway, a donation of electrons for bonding power, and you're going to generate glyceraldehyde 3-phosphate and eventually a larger sugar, glucose. I do want you to think about CO2. It's nice in this picture this way. CO2 and the sugar being bookends of one another. Notice the little gray circle here represents carbon, and carbon is in your sugars, and most of our macromolecules are grounded or carbon-based molecules. So carbon dioxide serves as the basis to build your sugar around. And then conversely, when you break apart the sugar, when we eat food and we are breaking apart sugars in our body, what are we breathing out? CO2. So when you're building a sugar, you need CO2. Where do the carbons come from that are in the sugar? C6H12O6. Where are the carbons coming from? Well, they're coming from carbon dioxide. Now let's look at it from the opposite way. When you're eating sugar, when you're eating pizza dough, when you break apart those carbon-based molecules, what are you going to break it down as and release it out as carbon dioxide? So they're kind of bookends of one another. But for the purposes of photosynthesis, CO2 is the kind of base molecule to build your sugar around. And that's the light independent reaction. And here, ATP and NADPH, those were your products from the light dependent reactions. Okay, let's take a look at the multiple choice questions, or a collection of the multiple choice questions. A scientist states that the incorporation of CO2 into organic biomolecules during the process of photosynthesis does not violate the second law of thermodynamics. Which of the following best justifies the scientist's assertion? All right. I love questions like this because the answer choice should mirror or be synergistic with, or should be in line with what the scientist has already asserted. Okay. The activation energies of chemical reactions are lowered by an organism's enzymes. Ecosystems are formed by interactions between a community of organisms and their environment. The entropy of the total system, which is the sun and all of the photosynthetic organisms, increases over time. The organic molecules created during photosynthesis are used for growth and repair. So you're going to be answering your questions in Edpuzzle. So I'm going to give you a minute now to go ahead and log your answer choice. All right, what did you think? What did you think? I want you all to be very, very careful really quickly. I'm not going to do this for all of the questions, but I'll do it for this one. Be very careful of answer choices that appear or in your gut may feel like they're correct, but they're not in line with the scientist's assertion, because it asks us which of the following best justifies the scientist's assertion. So the scientist is talking about the second law of thermodynamics, and that we're not violating the second law of thermodynamics. So the answer choice should be consistent with what the second law of thermodynamics says. So that means that the first answer choice, the second answer choice, and the fourth answer choice are out because they have absolutely nothing to do, in a very clear sense, with the second law of thermodynamics, which states that with each energy transformation or metabolic process, you're going to increase delta S or entropy or disorder in the the atmosphere around you, the organism. Okay. So the correct answer here is the entropy of the total system, the sun, and all of the photosynthetic organisms increases over time. All right. And then you see here captured at the bottom, kind of a summation of that second law of thermodynamics again. Every reaction releases heat, which increases the entropy of the entire system, even though the reaction in question creates more ordered products. All right. What we mean by the entire system is everything, you and everything outside of you. All right. Let's take a look at multiple choice question two. Me and my cursor. A scientist asserts, remember, I love these questions. A scientist asserts that spinach chloroplasts capture the most light energy from waves within wavelengths from 500 nanometers to 600 nanometers. To evaluate this assertion, the scientists placed spinach chloroplasts in separate chambers and exposed them to different wavelengths of light. For each chamber, the scientists measured the amount of gas, or O2, that was produced in an hour. Which of the following best represents data that supports the scientist's claim? So here again, you're looking at the four graphs here, and you're going to select the graph that best match what the question already is telling you. So which graph matches what the scientist already believes? Go ahead, use Edpuzzle to log your answer choice. Nice. So how'd you do? How'd you do? Sweet. All right. This graph shows that the most oxygen produced is produced when the leaves are exposed to light in the 500 to 600 nanometer range. Right. So this is the graph that you're looking for right here. And here they're using oxygen production as being a kind of proportional relationship to the amount of light that the plant is being exposed to, the amount of light that's coming in, right? Because you remember, that's when you're going to be producing oxygen when the light dependent reaction is active, getting a lot of sunlight. All right, let's take a look at question three, multiple choice three. Let me move myself out of the way so that you can see the answer choices. There you go. Whee! All right. So it says a scientist asserts that the appearance of atmospheric oxygen, which occurred around 2.3 billion years ago, resulted from the activity of prokaryotic organisms. The claim is based upon the evidence present in the figure included below. Which of the following best supports the assertion? So be clear about the scientist's assertion in the question setup, and then find which answer choice best supports that assertion. And also, when you have pictures or diagrams or graphs in the question, be sure to try to understand as much about them, because most often your answer can be supported by the information here. So make sure that your answer choice is consistent with this diagram as well as the picture that's set up here. Go ahead and log your answer in Edpuzzle. Okie doke. Let's see. Let's see the correct answer. The correct answer here is, let me move myself again. The correct answer is evidence that the first photosynthetic bacteria produce oxygen gas as a product of photosynthesis. Cyanobacteria release oxygen into the atmosphere as a product of photosynthesis. Before the rise of cyanobacteria, there was no atmospheric oxygen gas. Super important, super important. All right, question four. Look, I'm not in the way this time. A scientist claims that an herbicide slows plant growth by inhibiting the function of chloroplasts. To test this assertion, the scientists exposed chloroplasts to increasing concentrations of the herbicide. The data are included in the table below. So let's look at the data table, use it to your advantage. You got the herbicide concentration, which is increasing. pH difference across the thylakoid membrane. Think to yourself where we talked about pH difference, when we talk about acid base. You see that difference? And then you see ATP production level. Let's look at the answers, possible answers. The herbicide allows more carbon dioxide to cross the membrane and lower the pH within the thylakoid. ATP synthase activity depends on a hydrogen ion gradient. The herbicide catalyzed the hydrolysis of ATP, or the herbicide increased the production of pyruvate and acid. Go ahead and take a minute in Edpuzzle and log your answer choice. Okie doke. Let's just process the table really quickly. So the scientist believes that the herbicide is inhibiting the function of the chloroplast. Let's see in what way. So as you increase the amount of herbicide, what's happening to the differential of the hydrogen ions that build up in that thylakoid space? It goes from very high to very low. And remember, in the light dependent reactions, if you don't get that high on the inside, low on the outside of those H plus ions, you'll never have the high enough differential for that proton motive force to go through ATP synthase to generate ATP. And so for this question, the correct answer choice is ATP synthase activity depends on the hydrogen ion gradient. The hydrogen ion gradient. Let me move myself again. ATP synthase must have a proton gradient in order to synthesize ATP. The herbicide appears to inhibit the creation of a proton gradient. Do not let the test fool you. If they're talking about acidic or basic or pH difference around photosynthesis, most often they're going to be talking about high hydrogen concentration, which is acidic, versus low hydrogen concentration, which is basic, which is pH. Don't let them throw you off. You're smarter than the test. All right. Let me move myself down here. Let's look at question 5. Question 5. Why won't they let me try up? All right. All right. Cyanobacteria contain a group of accessory pigments and proteins called phycobiliproteins. Green algae lack these proteins. Okay, so green algae do not have them. A scientist claims that the phycobiliproteins allow cyanobacteria to survive in some aquatic environments better than green algae. Which of the following justifies this claim? So again, zero in on what the scientist is trying to say, and then find the answer choice that is most synergistic. Is it the pigments allow the cyanobacteria to store light energy so that it can be used for the dark, after dark for photosynthesis? The pigments can be used as an energy or food source when there's not enough light to carry out photosynthesis. The pigments absorb wavelengths of light that green algae cannot absorb. This allows the cyanobacteria to capture more energy than green algae for photosynthesis in some environments. Or the pigments block light from passing through the deeper water levels and therefore block the growth of green algae. Go ahead and log your answer in Edpuzzle. I almost feel like I should be playing the Jeopardy music. All right, these should be getting a little easy for you now, a little easier to kind of work through now. All right, the correct answer here is the pigments absorb wavelengths of light that green algae cannot absorb. This allows the cyanobacteria to capture more energy than green algae for photosynthesis in some environments. And remember, the pigments allow cyanobacteria to absorb wavelengths of light, the sources of energy that green algae cannot absorb. So you got to really lock into the function of pigments. That kind of leads you to the correct answer. All right, let's quickly talk about cellular respiration. We don't have time to cover all of the details of cellular respiration, but we're gonna get some high points. We're gonna look at a few multiple choice, and then we're gonna look at some FRQs. Okay. All right. So fermentation and aerobic or aerobic respiration are metabolic pathways that allow organisms to use energy stored in organic molecules. In other words, the molecules that are produced by autotrophin producers at the end of photosynthesis, these sugars. Cellular respiration is the process that's going to be able to basically oxidize or bust up or break apart those sugars and convert that to energy we can use, which is ATP. This is I want to be very clear about this point. All living organisms, most all living organisms will have to do cellular respiration in part or in totality, including plants, including photosynthetic organisms. Photosynthetic organisms can't just make sugar. They also have to break down that sugar for energy. So they have to do photosynthesis and cellular respiration. For you and I, no photosynthesis, but cellular respiration, okay? All organisms carry out fermentation and or aerobic cellular respiration, as I just said. Fermentation and aerobic respiration transfer the chemical energy stored in the bonds of organic food molecules like sugar. It can be, remember, it can be proteins and it can also be lipids and convert and transform that energy into ATP, or convert that energy into ATP. ATP is used to power cell work. And we know why. We took a look at the structure of ATP and we know why. It's high energy, it's small, it's easily accessible. Aerobic cellular respiration requires oxygen as a reactant and releases carbon dioxide as a product. All right. And you'll see when we look at the reaction, oxygen's job in cellular respiration is to be the final electron acceptor. So as you break apart, remember, you put electrons into photosynthesis to build a sugar. In cellular respiration, you're going to extract electrons as you break down the sugar, and you're going to use those electrons. But who's going to be holding the electrons at the end of the day in cellular respiration? That's going to be oxygen. And that's why oxygen is so important. That's why we need it to live, because it is the final electron acceptor in cellular respiration. Okay. And you're going to release carbon dioxide as a product. We can't use carbon dioxide. Photosynthetic organisms can. All right. Another important thing I want to mention here. Sometimes students hear that cellular respiration and photosynthesis are reverse reactions. It is that the products of one are the reactants. They're very similar. Reactants of the other, the products of one are the reactants of the other, but they are not reverse reactions. You can't run cellular respiration in reverse and do photosynthesis and similar with photosynthesis. All right. Light is required for photosynthesis. Is a very nuanced process and it's a wildly different process, uses different enzymes. They're not reverse reactions. Okay, so that's important. Okay. All right. Aerobic cellular respiration, like photosynthesis, uses chemiosmosis to manufacture ATP. So remember that passing of electrons to create that proton gradient, the use of ATP synthase, where you've got high hydrogen concentration versus low on another side of a membrane, and using that actual rotary protein to phosphorylate ADP into ATP. It is very similar to what we've talked about already in photosynthesis. That's how we make a large amount of ATP in cellular respiration. Aerobic respiration is much more efficient than fermentation or anaerobic respiration. Here's a quick snapshot of the two processes, just really quickly. And we're going to simplify this by using the 6-carbon molecule glucose. The first process of glycolysis, you're going to need a net input of ATP to get that thing started. Without the presence of oxygen, you'll be in the fermentation route. You're not going to make near as much ATP. With oxygen present, you will do glycolysis, the Krebs cycle, and the electron transport chain. And we already know how this electron transport chain is going to work. It looks a lot like the light-dependent reactions of photosynthesis. But the point is, you're going to make a large amount of ATP so that living systems can do cell work. All right, and here are those stages captured again for aerobic respiration. I'm going to try to be efficient here. Here's glucose. Remember, I told you, connect glucose with CO2. And here we're going to be oxidizing glucose or breaking glucose apart. Okay, so the first step is to break glucose into pyruvate molecules. When you break apart a molecule, you extract electrons off bonds. You're going to give those electrons to the electron acceptor. That is a redox reaction, okay? Similar to where we did NADP plus to NADPH in photosynthesis. Here, you're going to reduce NAD plus to NADH, Okay, so here's the rub. Every time you break off carbons or break apart or rearrange this glucose molecule, you're going to be extracting electrons out of it. Those electrons are going to go to these electron carriers, NADH, and FADH2. Where are they taking the electrons? They're going to take the electrons to the electron transport chain. Now, you do make some ATP along the way, substrate level process where it's an enzymatic process. So some low levels of ATP made there, but you get your big bang for your buck here with oxidative phosphorylation, and that happens in the electron transport chain. Notice our location. Glycolysis happens outside of the mitochondria, and here this is another membrane-bound organelle. Here, the mitochondria, this is where you'll have the citric acid cycle or the Krebs cycle and the electron transport chain occur. And these folds here are membranous, very similar to what we see on our cell membranes, very similar to what we see in the thylakoid and on the outside of the chloroplast. So there's a lot of kind of consistency across these structures, but just know that these processes will use these membranes for the process, in particular, the electron transport chain. All right, so we're going to zoom in on the electron transport chain, okay? Remember, CO2 and the sugar are connected. So you start to break apart and rearrange the sugar. As you did that, you release those carbons as CO2, but what was important to you? You needed to extract the electrons from that sugar, and that's exactly what you did. You got all of the electrons from the sugar, or most of them. You donated those electrons to the electron carriers, NADH and FADH2, and they brought those to the final stage of aerobic cellular respiration, the electron transport chain. Okay, and this process is generally called oxidative phosphorylation, primarily because it's a series of redox reactions and oxygen is going to be your final electron acceptor here. Okay, so NADH and FADH2 are going to drop off their electrons at different spots here in the electron transport chain. This should look similar to you. You've got a sequence of proteins that are slightly more electronegative as you go along. So they're calling for the electrons. They have a pull for the electrons that are being dropped off. So as the electrons move, notice that the hydrogens are being pumped from this mitochondrial matrix, this mitochondrial matrix to this inner membrane space. That is creating the high-low with hydrogen that we've seen before. We saw it in the light-dependent reactions of photosynthesis. The movement of the electrons themselves along this golden line, the electrons are coming, you see them here. When you see the electrons as they're moving, they're going to, because of the charge difference, you're going to see that's going to stimulate the movement of the hydrogen ions from the mitochondrial matrix out into the inner membrane space. High hydrogen, low down here in the mitochondrial matrix, we don't like that. So the hydrogen ions will barrel back through ATP synthase. This protein will physically turn, and that energy generated is going to phosphorylate ADP to ATP, or adenosine triphosphate. And so you see that process, or that word again, chemiosmosis. Okay, so this looks very, very similar to what we saw in the light dependent reactions of photosynthesis. I'm trying to see if there's anything else super important here other than in prokaryotes. Remember, they don't have mitochondria, but they do have a cell membrane that looks a lot like this. So this process will occur there. And so if I go to the picture just before, this is where we were. This is where we are right here. Okay, you've extracted the electrons from the food, from the sugar, and you've used those electrons to create a proton gradient, high and then low. And it's that proton gradient, that's what you absolutely need to barrel back through ATP synthase, this protein here, to phosphorylate ADP to ATP. And remember, what I mean by phosphorylate is add a phosphate group to ADP, adenosine diphosphate, to adenosine triphosphate. All right, let's look at multiple choice question 6. Nearly all organisms on the earth perform glycolysis. How does this support the claim that glycolysis is one of the oldest metabolic pathways on earth? Let's look at the answer choices. To be present in almost all organisms, glycolysis was most likely present in one of the common ancestors of all living things on earth. Glycolysis is a recently evolved pathway that has been widely used because it's so beneficial. Because glycolysis is absent in few higher organisms, it can't be one of the oldest metabolic pathways. Or the last answer choice, because there are organisms that do not do glycolysis, it can't be one of the oldest metabolic pathways. I'll let you log your answer choice now in Edpuzzle. How y'all feeling? I know y'all rocking these multiple choice. I can feel the energy. I can feel the energy. I know you're doing well. Okay. All right. So it's the question says nearly all organisms on Earth can perform glycolysis. Okay. And so that takes at least one answer choice out of the mix altogether. Okay. So it seems like glycolysis is a highly conserved process. And so the best answer choice here is the first answer choice. To be present in almost all organisms, glycolysis was most likely present in one of the common ancestors of all living things on Earth. Glycolysis is present in nearly all organisms because it was present in the common ancestor of all these organisms. It's a highly conserved process. Let's take a look at question 7. In the electron transport chain of cellular respiration, blank is or are oxidized, and the energy from this oxidation is used to generate blank, a blank gradient. Let's look at the answer choices. Carbon dioxide, then hydrogen ions, pyruvic acid, then electron, NADH and FADH2 and hydrogen ions. And the last one, electron carriers and electrons. So use Edpuzzle to figure out the fill in the blank for the first blank and the second blank. Go ahead and log your answer now. This one can be a little bit tricky, but I think you all got it. In the electron transport chain of cellular respiration, NADH and FADH2 are oxidized, and the energy from this oxidation is used to generate a hydrogen ion gradient. So the correct answer, let me get my cursor, is NADH, FADH2, and H plus ions. I'm going to go back to the picture really quickly. This one, right? We talked about this in photosynthesis, but when these molecules give up their electrons, they are oxidized, right? When they give up their electrons, they are oxidized, all right? And so here you can see here, I think I failed to, I mentioned this, but not with the picture, that oxygen is the final electron acceptor. And when oxygen gains those two electrons with the hydrogen ions that are now in this mitochondrial space, water is also a product of cellular respiration. That's really important. So I needed to come back here. Okay. So remember, where do the electrons come to at the end of the day? Back to oxygen here. It's kind of a full circle moment for water and oxygen gas as well, because if you go back to photosynthesis and the light dependent reaction, we had to bust up water, right? And then when we bust up water, we extracted the electrons there, all right? And then oxygen gas was a byproduct. Here, we have oxygen gas. The oxygen gas receives the electrons, and then with the hydrogen ions, you now can construct water. So think of CO2 and the sugar as bookends of one another. Think of water and oxygen gas as bookends of one another across photosynthesis and cellular respiration. Hopefully that makes sense. If not, your course instructor will definitely be able to support you. All right, let's look at our last multiple choice question. A chemical makes the mitochondrial membranes permeable to hydrogen ions. Which of the following is most likely if the chemical is added to cells? The cells will make less glucose. The cells produce less ATP. The mitochondria need more glucose to generate the same amount of ATP. The cells will produce more ATP. Go ahead and log your answer choice in Edpuzzle. All right, how'd you do? The correct answer choice here is the cells would produce less ATP. ATP synthase must have a hydrogen ion gradient to produce ATP. The chemical will inhibit the creation of the hydrogen ion gradient, therefore inhibiting the creation of ATP. Let me go back to the picture to nail this one, okay? It says the mitochondrial membrane is permeable or leaky. The mitochondrial membrane is leaky to hydrogen ions. Now watch this. Let's go back. Let me go back to this picture here. All right. So this process here is going to be happening on these little folds here. They're called cristae, the folds on the inside. If the mitochondrial membrane is leaky to hydrogen ions, that's going to prevent you from building the gradient on the inside of the mitochondria. And so the question is saying this outer membrane is leaky. Okay. And so when you get to here, you won't be able to build this gradient because you're losing hydrogen ions on the outside membrane of the mitochondria. And I hope that makes a little bit of sense. All right, so that was multiple choice 8. All right, let's take a look at the relationship between photosynthesis and aerobic cellular respiration. And I've talked about this a little bit already, okay? The first thing, the equations. Students will also a lot of times want to say that these are reverse reactions. They absolutely are not. And I kind of land on that. Sunlight is required for photosynthesis. There are different enzymes used in the process. They're in different places. Chloroplast versus mitochondria, they are happening in wildly different places. But what is similar are the actual molecules themselves, all right? So you see that CO2 and water are the reactants for photosynthesis, and you'll see that CO2 and water are products, along with ATP, of cellular respiration. Okay, and then you see the products of photosynthesis, which is a sugar here, glucose, and oxygen gas, are the reactants for aerobic cellular respiration, glucose and oxygen here. So they do use the same molecules, but they're not reverse reactions. Another thing to note here, that you have to have energy into photosynthesis. Having plus energy here is probably a little less consistent. A lot of times I'd like to see like energy written on the arrow just because energy itself is not, made of like, it's not matter, it's photons of light. You can't find it in the periodic table, but either way, that's not a huge deal. Like you can just write sun on this arrow right here would probably be better representative of what's happening, okay? But energy in, so this is an endergonic reaction. This is anabolic. You're building a larger molecule. So you're taking small CO2 molecules. Remember, that's the backbone of your sugar. And you're going to build your sugar around carbon dioxide. Where do you think the carbons come from that are here? They come from here. You have to have a net energy in to build that large sugar. That comes from sunlight energy. This is an anabolic overall, an anabolic process. If you look at aerobic cellular respiration, you're going to be oxidizing or breaking up that sugar, oxidizing, breaking it up, okay? And remember, in catabolic reactions, when you break them apart, most often you'll get a net energy out or exergonic reaction. And so that's what you see here. The energy out here is appropriate in this equation because it's an actual molecule, it's ATP. It's ATP, okay? And this picture is a nice kind of summation of the flow of energy, which we talked about at the beginning, and the cycling of the products and reactants of each process. All right, let's look at our first free response question. Okay, let's end by answering 2 FRQ questions. Let's read this one. Researchers hypothesized that the plant compound resveratrol improves mitochondrial function. To test this hypothesis, researchers dissolve resveratrol in dimethyl sulfoxide, or DMSO. The solution readily passes through cell membranes. They add the resveratrol solution to mammalian muscle cells growing in a nutrient-rich solution or culture medium that contains glucose. Glucose is important. They measure ATP production at several time points after the addition of the resveratrol solution and find an increase in ATP production by muscle cells. So for your answer choice that you'll answer in Edpuzzle, you need to describe the primary advantage for mammalian muscle cells in using aerobic respiration over fermentation. Then identify an appropriate negative control for this experiment that would allow the researchers to conclude that ATP is produced in response to the resveratrol treatment. C, to predict the effect on short-term ATP production when resveratrol-treated mammalian muscle cells are grown in a culture medium that lacks glucose or other sugars. And finally, the researchers find that resveratrol stimulates the production of components of the electron transport chain. The researchers claim that treatment with resveratrol will also increase oxygen consumption by the cells if glucose is not limiting. Justify the claim. All right, so for this question, you need to describe, identify, predict, and then justify. Make sure that the answers that you log in Edpuzzle rise to the verb demand that each subpart is requiring. So go ahead, take a moment, take a couple of minutes, and go ahead and write out your answer to FRQ 1. All right, let's take a look at the scoring rubric. Well, it's not really a rubric, but the scoring kind of guidelines for FRQ1. Okay, so there's the question kind of captured again. Let's look at A part. Describe the primary advantage for a mammalian muscle cell in using aerobic respiration over fermentation. That's pretty straightforward, and we had a slide on that one, right? More ATP per glucose molecule is produced by aerobic respiration. So, in the presence of oxygen gas, you can do glycolysis, the citric acid cycle or the Krebs cycle, and the electron transport chain. So, you can do all three phases and produce a large amount of ATP. Let's look at B. Identify an appropriate negative control for this experiment that would allow the researchers to conclude that ATP is produced in response to the resveratrol treatment. Okay, and so the readers will accept one of the following. The researchers must run the experiment without resveratrol, or the researchers must treat the cell with the dissolving agent or the DMSO alone. Okay. And so those would be two acceptable negative controls for this particular experimental setup. C, predict the effect on short-term ATP production when resveratrol treatment mammalian muscle cells are grown in a culture medium that lacks glucose or other sugars. Two answer choices could be accepted here: no ATP production or a reduction in ATP production. And that one is pretty straightforward as well. Let's look at D. The researchers find that resveratrol stimulates the production of components of the electron transport chain. The researchers claim that the treatment with resveratrol will also increase oxygen consumption by cells if glucose is not limiting. Justify the claim. And the answer here is more electrons can be transferred so that more oxygen is required as a final electron acceptor. All right, you're going to need more oxygen so that you can receive the electrons in the electron transport chain. I think that this FRQ1 was pretty straightforward. And if you just capture the information that we talked about with cellular respiration in this session, I think you would do solid. You would do a bang up job and probably get all of the points for this particular question. Okay, now let's take a look at free response question 2. It says non-cyclic electron flow and cyclic electron flow are two major pathways of the light-dependent reactions of photosynthesis. So we're back on photosynthesis. In non-cyclic electron flow, electrons pass through photosystem 2, then components of a chloroplast electron transport chain, and then photosystem 1 before finally reducing NADP plus to NADPH. In cyclic electron flow, electrons cycle through photosystem 1 and some components of the electron transport chain. So in non-cyclic flow, you should be familiar with that. That's the one we talked about already. And then here they have bolded the cyclic flow. They have it labeled and bolded. Okay, so let's take a look at the questions that go along with this particular figure. And you can see a description of the figure below. Anytime you see figure descriptions, make sure you read those as well. So here are the questions that go along with free response question 2. A, describe the role of chlorophyll in the photosystems of plant cells. B, based on figure 1, explain why an increase in the ratio of NADPH to NADP plus will cause an increase in the flow of electrons through the cyclic pathway. C, using rice plants, scientists examine the effect of a mutation that results in the loss of the protein CRR6. CRR6 is a part of the photosystem 1 complex, and its absence reduces the activity of photosystem 1. Predict the effect of the mutation on the rate of biomass or dry weight accumulation in the plant. And D, justify your prediction in part C. So try to be efficient here. If you want to take a screenshot with your phone or something like that to capture the questions for free response question 2, and I go back to the figure so you can look at that while you're constructing your answer for Edpuzzle. If you think that would work, go ahead and take a minute, take a screen capture or screenshot with your phone of the questions here, and I will toggle back to the figure for a minute and give you a minute to answer those questions in Edpuzzle. Just showing you the questions again here. Let's go ahead and take a look at acceptable responses to FRQ2. All right, let's move myself again. I'm doing lots of, hopefully I'm not making you all disoriented by all my movement. Okay, doke. Let's see, move me over. Okay, so A part. Describe the role of chlorophyll in the photosystems of plant cells, except one of the following. Chlorophyll captures or absorbs light energy. Got that. Chlorophyll receives electrons from water, or receives electrons from an electron transport chain, or transfers electrons to an electron transport chain. Either one of these answer choices or a combination of these answer choices would be accepted and would get you 1 point. Okay, I think that was pretty straightforward. I'm sure all of you got that one. Let's look at B. Based on figure 1, explain why an increase in the ratio of NADPH to NADP plus will cause an increase in the flow of electrons through the cyclic pathway. Now, think about in your head what happens to the electron in the non-cyclic pathway. The electrons end up with NADPH, and NADPH carries those electrons to the light-independent reaction so you can build your sugar, bonding power for your sugar. But if you increase the ratio of NADPH to NADP plus, that means you have all of your electrons locked up in NADPH. Remember, NADPH is your with electron form. NADP plus means that you can accept electrons. So this is the one that's carrying the electrons. This is the one that will receive the electrons. So if you have this, an abundance of this, or a high ratio of NADPH, well, there's no place to give the electrons to go to the light independent reactions, which means you're going to have to utilize that cyclic pathway a little bit more than perhaps you might want to. All right. And I'm personifying a little bit there, but you get the point. Okay. And so the correct answer here would be there is less or no NADP plus to accept the electrons. So the electrons pass instead to the cyclic pathway from ferredoxin to the cytochrome complex. Right. And this is a pathway for these plant cells to make ATP. If they get in a rut in ATP and they need some ATP, this is a pathway to which they can do that. All right, let's look at C. Using rice plants, scientists examine the effect of a mutation that results in the loss of the protein CRR6. CRR6 is a part of the photosystem 1 complex, and its absence reduces the activity of photosystem 1. Predict the effect of the mutation on the rate of biomass or dry weight accumulation in the plant. The answer here, the acceptable answer here is the rate of biomass accumulation or biomass will be lower in comparison with plants without the mutation. All right, so a decrease in biomass. As you don't have one of your photosystems functional, you're never going to be able to build biomass, which happens in the light independent reactions, which is building your sugar. Your sugar is the primary kind of biomass of the plant itself. Now let's look at D part. Justify your prediction for part C. And that answer is there will be insufficient ATP and NADPH produced for the synthesis of carbohydrates in the Calvin cycle, which I kind of just stole my own thunder, but that answer is consistent. All right, so my justification for C was the answer for D. All right, And yeah, so that's FRQ2, the scoring guidelines. And that was a total of 4 points on that one. So thank you all so much for joining me in the bioenergetics study session, AP bio study session. And I hope to see you all soon. Maybe I'll see you in person, a live Zoom session, or in another recorded session soon. I'm Paulette Evans.